lays a single egg. And the males, if you zoom in here, they fight for access to the females. So, and there are two different morphs. There are large horned morphs and small horned morphs. That is a complete nutritional polyphenism. So a well-fed larvae will develop into a large horned male, whereas a small, poorly fed larvae will develop into a small horned male. And they have different reproductive tactics. So the large horned males fight for access to females, whereas the small horned males sneak copulations. So the three species that I chose represent diversity at a number of levels. So not only in the size and shape of the horns across the species that are generally expressed in the males. So in this Nigriventris species, you see this dramatic thoracic horn that can be as long as the body length of the animal. In Taurus, you have head horns that show this beautiful bowl-like shape. And then in Sagittarius, you have these relatively minor head horns. They also differ in the degree of the nutritional polyphenism. So Nigriventris has a really dramatic nutritional polyphenism, very nutritionally sensitive, and followed by Taurus, which is nutritionally sensitive but less so. And this has been pretty much lost in Sagittarius, where a small male just looks like a smaller version of a large male. And they also differ in the degree, or the polarity of the sexual dimorphism. So across most of the 2,400 species, males have the horns, but there are a few rare exceptions, like the Sagittarius here, where the females have the more dramatic horns compared to the males. So they represent diversity, they have diversity across a number of axes. And they're closely related as such. So Sagittarius and Taurus shared a common ancestor about 5 million years ago, and they shared a common ancestor with Nigriventris about 15 million years ago. And predictions for genes to be involved are hox and limb patterning genes, genes related to growth and metabolism, and genes that show sex specificity based on a number of different studies that we've conducted in the lab. So to address this question, to try to identify the genes that might underlie this phenotypic diversity, I took a comparative transcriptomics approach and compared and used 454 sequences or transcriptome assemblies from Taurus and Nigriventris, and made one for Sagittarius with Illumina sequencing, and identified some 17 and 23,000 of the breeding frames for each of the species, and then whittled that down to about 2,000 high-quality orthologous gene sets with one member of each gene from each species. And then for each of those orthologous gene sets, I can calculate rates of functional evolution, and I'll tell you how I did that in a second. With these rates of functional evolution, I can integrate those data with gene function and gene expression data to address these two following questions. So are there specific functional classes of genes that are targeted by selection? And are the genes that drive these within species polyphenisms, those are the same as those that drive divergence between species? So for the first question, the metric of functional sequence divergence that I used was DN/DS. And as a refresher, DN is a measurement of protein function evolution, so the number of amino acid substitutions weighted by the number of amino acid sites over the number of synonymous substitutions per synonymous sites. So as you can intuitively see, looking at this ratio of ratios, the DN/DS value greater than 1 would be indicative of positive selection or functional evolution. But as you can also imagine, you can have one important amino acid substitution that could be swamped by three synonymous substitutions, and therefore have a really low DN/DS value. So this is an excellent opportunity to integrate multiple genome-wide data sets to test for signals of selection, particularly when the traits that you're interested in might be complex phenotypes and may in fact require the action of multiple genes interacting. So here is the DN/DS distributions for the three different species pairs. And I'll just show you the whole distribution, which is actually very typical for, very similar to other species. It'll just draw your attention to the tail here. There are just 16 genes with signals of positive selection, so DN/DS values greater than 1. And what are those genes? Well, a number of them are particularly interesting for functional studies. So there's ADAM-TS3, which is extracellular matrix protein, important in developing connective tissues. There's FGF, the fibroblast growth factor receptor related protein, which is important in angiogenesis and wound healing and development. And the nodal modulator protein, which is a collagen enzyme that has been shown to be important for normal epidermal tissue development. So these are top candidates for functional studies that are underway. So if I zoom in on this DN-DS distribution here, so we're looking now, the scale is just to 1, and we're looking at the 3 species pairs. So the most closely related species pairs, Sagittarius and Taurus, are in bright green. The pair with the broadest distribution is Nigriventris and Taurus here. So these two with a similar degree of nutritional polyphenism. So again, this is a case where you can use functional enrichment to see if there are any classes of genes that are standing out as diverging more than expected by chance. So to do that, I characterized the protein function for all of them and tested for elevated divergence in each functional class of genes. So for this pair, Nigriventris and Taurus here, that has the broadest distribution, I found no signature of functional enrichment. So these high DN-DS values, so the broader distribution, was actually random with respect to protein function. In the Nigriventris Sagittarius pair, there were 2 categories showing enrichment both related to metabolism. And finally, in the most closely related species pair, Sagittarius and Taurus, where there's been the loss of the nutritional polyphenism and the reversal of the sexual dimorphism, I found 23 categories enriched for divergence. And if we look at what these categories are, 15 out of the 23 categories are related to metabolism, and another 6 categories are related to biosynthesis. So this is a really cool result, particularly for this species pair, because there's been this loss of the nutritional polyphenism. So we're seeing faster evolution in metabolic genes in this species pair where there's been this nutritional polyphenism loss. So are there specific functional classes? Yes, in just the most closely related species pair and in metabolic and biosynthesis genes. And for this next question, to understand if the within-species polyphenisms might be related to between-species divergence, I used a large-scale microarray study that we generated measuring gene expression patterns within this Taurus species. And so I characterized genes as nutritionally sensitive, differentially expressed between the sexes, or differentially expressed between tissues by comparing large versus small males, females versus males, and head horns versus thoracic horn tissues. And then tested for elevated DN-DS in each of these functional classes of genes defined based on gene expression within a species. So here are these 3, and I'll summarize these results with these 3 different classes of genes that vary in expression within a species. And across the top will be the 3 different species pairs. And I'll put the predictions with X's and check marks based on their morphology, what we would expect to see, and the results with asterisks and non-significant. So for this Nigriventris Sagittarius pair, this is here with the nutritional, with so they differ in the degree of nutritional sensitivity. So you would expect to see a signature of divergence there. They also differ in the polarity of the sexual dimorphism, and there's not a clear prediction based on the position of horns where you would expect to see difference. And here's what we find. So this does not match predictions, so I would have expected to see a signature of selection there. This does match predictions, and this, by default, I guess, matches predictions. So then in the Nigriventris and Taurus pair, where they have different degrees of nutritional sensitivity, we see a signature that matches predictions here. They have the same polarity in the dimorphism, the sexual dimorphism, and they have a difference in the position of the horn. So in Nigriventris, it's the dramatic thoracic horn. In Taurus, it's the head horns. So we see a really strong enrichment in genes that are differentially expressed between tissues. And finally, in the most closely related species pair, they are different in the degree of nutritional sensitivity, also different in the polarity of the sexual dimorphism, and they also have different positions of their horns. And we see the strongest signatures of selection, again, in this most closely related species pair, with similar mechanisms that, with enrichment in genes that drive the differentiation within a species. So these results, these patterns match the predictions that we would have based on morphology. And to summarize this, we see rapidly evolving genes that have DNDS ratios greater than 1 in genes related to growth and cuticle development. We also see rapid evolution in metabolic and nutrition-sensitive genes in this most closely related species pair where there's been this loss of the nutritional polyphenism. And finally, in the last part that I showed you, that we see that there are similar mechanisms that may be promoting diversity both between morphs and sexes, as well as showing signatures of divergence between species. So finally, one of the projects that I won't have time to tell you about today, but is doing population genomics of this Taurus species, which is native to the Mediterranean, but was deliberately introduced to Australia, and then maybe deliberately introduced to the US. Farmers like them because they clear away the dung and reduce pests. But it was not supposed to be a legitimate introduction. But these three populations in just 50 years have rapidly diverged, and the introduced populations have diverged in opposite directions from the native population. So in Australia, you have to be a very large-bodied male before you're going to have large head horns. In contrast, in the US, you can be a very small-bodied male and have head horns. So there's lots of tiny males walking around with big head horns in the US compared to Australia. I'm sure Australians would like to hear that. But, so I can't give you the punchlines here yet, but stay tuned. And with that, I hope that I've shown you that we can get at some of the power and the promise of genomics. So there are a number of functional classes of genes that we would have expected to show signatures of divergence or signatures of selection underlying these complex phenotypes. In urchins, those related to ion homeostasis, lipid metabolism, and biomineralization. And in the beetles, those related to metabolism and response to nutrition. And there were also a number of functional classes of genes that we wouldn't have necessarily, that we identified that we didn't necessarily predict ahead of time. Those related to cell-cell adhesion in urchins and biosynthesis in the beetles. So the take-home messages that I hope you'll have from this talk are that purple sea urchins may be able to adaptively evolve in response to future climate conditions, and that there may be shared mechanisms that promote diversity both within and between species of beetles and maybe more generally as well. So with that, I'd like to thank you for your attention and thank my mentors in this research, Armin Wojcik, Steve Columbi, and Eric Sanford, and Doug Emlin and Ian Warren for sharing with me unpublished data, and Carrie Ganote and James Peace, both at Indiana University, NSF for funding the postdoctoral fellowship that supported the beetle research, and the National Center for Genome Analysis support at Indiana University, and then as well as a number of people that have helped along the way in one capacity or another. And I'd also like to emphasize that I'm now very close by, so that's a nice connection. And if you or anybody you know shares some of these research interests, I'll be accepting PhD students in the fall and hiring a postdoc and technician in the next 6 months. So there's my shameless plug. So thank you all for your attention. I'll be happy to take questions.